when it sends its action potential down, that spreads out along all these branches and contracts all those muscle fibers. So we're going to step back from that for a minute and look at that response to a single action potential in a little bit different way than we did previously, right? So we're going to look at a twitch contraction. What we're going to look at here is this myogram over here that's measuring the force of contraction as a function of time, right? Last time we kind of looked at the force of contraction as a function of the starting length, right? We had the 100% optimal length over here, and then to the left and right of it was suboptimal length. This time we're looking at the time sequence from the nerve impulse as it travels down the sarcolemma, and as that sarcolemma releases the calcium and the cross bridge formation happens, and till the time when that signal, the calcium starts being pumped back in. Right, so if we look at this chart over here, we see this latent, well, we see the nerve impulse first, right? That's your starting point. The nerve impulse, right? The action potential from the neuron at the neuromuscular junction, right? Excitation, right? Depolarization, excitation. It happens, all those voltage gated channels open up down the length of the whole muscle fiber simultaneously, right? So as that's traveling right here, obviously it doesn't start contracting until calcium is released. So the period of time, the muscle action potential is sweeping down the muscle fiber, right? And the muscle hasn't contracted, doesn't know it yet. The sarcolemma doesn't quite know yet, or it just does, it opens up, but the cross bridge formation hasn't happened, right? Nothing happening. That's about a couple seconds right here. That's like 1 to 2 milliseconds, milliseconds, right? It's like bam, right? And then you get the process of the contraction and relaxation, which is much slower, right? So if this is 1 to 2 seconds, this is going to be like, depending on what type of muscle fiber, which we'll talk about later, this twitch is going to be much longer, right? So as the calcium is released, right, and the calcium is going to bind to troponin, myosin binding sites on the actin are exposed, cross bridges form, right? And then you get that tension, right? It's going to start contracting, shortening the sarcomere during this whole period. Right? And then at a certain point, calcium is being pumped back into the sarcoplasmic reticulum, and then the myosin heads released from the actin, right? And then tension starts to decrease. So think about, how we're looking at this for a second here. Right? Here is your sarcomere, right? Here's the stretched out one. This would be the contracted one, right? You have a signal, it's like, and then all of a sudden it goes, right? Contracts and then kind of goes slow down, right? That's the kind of timing you get. A quick action potential, calcium release, a slow kind of crunching of the sarcomere, and then a release like that, right? That's the timing that this is going to happen right here. Okay, so this is that whole graph, right? The muscle twitch, a myogram of a single action potential and what happens as a function of time right here. Okay, there's this relaxation period where it's going back to its resting length, right, from this contracted space. There's also a period, when you can't stimulate it again, but we're not going to talk about that until we get to the nervous system. Okay, we'll just stick with this kind of simple graph of how it contracts as a function of time after the nerve impulse. For that could be a single fiber, or because the motor neuron is innervating several fibers at once in that motor unit, right? This could be, a whole motor unit, right? Measuring a whole single, like a whole bunch of them at the same time. They would act the same in that motor unit. Individual fibers twitching in unison. So that's what those motor fibers do, motor units. All right, and then this also, just put a pin in this for later. Next, on Thursday, we're going to talk about metabolism and the different types of muscle fibers, fast ones, slow ones right here, right? And the name fast twitch, slow twitch, has to do with that myogram right there, right? How fast that action occurred, whether it was 20 milliseconds, 200 milliseconds. Right? So that'll be part of that. Don't worry about that right now, but just keep that in mind for later. So that was a single twitch. And now we're going to look at a couple of different things about how the force is going to be generated, not only in a single muscle fiber, but in a whole muscle, right? And what are the factors? Right? The frequency and the size and number of the motor units activated over here, okay? So the first one is this concept of stimulation frequency, right? And what its effect on the contractile force. Over here, right, we had that twitch. Right over here, single action potential, right? We can see that myogram right there, right? The little latent period, contraction, relaxation over here, right? This relaxation point. And then you see over here, this is just that you had another one and it was going up, but then before it has a chance to fully relax, you'll notice there's more little action potentials, right? Further stimulation over here. And then over here, you can see it rapid fire over here. So in other words, this muscle is receiving a whole bunch of input, right, signals from that motor neuron over here, right? And what we see here is that the force, right, is increasing as you move along here. So let's look at this graph a little bit closer. And so this whole thing right here, right, when you're looking at a graph like that, you're looking at the number, the frequency of the stimulation versus the force of contraction, right? We're thinking about when do you reach full so-called tetanus, that is maximum tension, right, with no relaxation. So let's look at this in a little more detail, right? You had a single action potential, and again, we have that single twitch myogram, right? Start up, contraction, up at this point, you start that relaxation phase, it goes down again. And then over here, you get another stimulation, right? It does the same thing, except right over here at this point, when it was about to go in its relaxation phase, it got hit with another action potential, which means it wasn't fully relaxed yet, right? It was in a partially contracted state. And because of that, you can generate more force. Remember, the length of the sarcomere, the starting sarcomere, was a factor in the force you can generate. So by this more frequent, right, a quicker stimulation before full relaxation occurred, you can get a greater amount of force by kind of summating, right? This that sums the two together. So you get this greater force from this level over here, whatever this force is, and then you're getting this much, right? So you could do the same thing over and over, right? Here it was going up, it was starting to relax, bam, gets hit with another one. And then as this one goes up, it was going down, bam, gets hit with another one. So it was in a more contracted state, you're generating more force, right? So each time you're increasing the force, right? You're actually keeping the calcium flooding the sarcomere, and you're just increasing that force right there, right? Keep on generating the contraction. You know, at this higher frequency, right? This 4 instead of 2, whatever, right? Giving it no chance to relax or little chance to relax, you get this sustained contraction, right? There's a little bit of a dip, right? Because it's not very fast. The stimulation isn't too much. So you get a slight chance to relax. So you get this kind of jagged graph looking thing, right? So this partial relaxation between stimuli, it produces a sustained contraction. It's a little bit wavering. It's not fully contracted. It's not a smooth, full contraction. If we increase the amount of stimulation as a function of time right here, right? At its highest frequency, the muscle never gets a chance to even relax. It all never goes back a little bit, and you get this, what's called tetanus, right? Maximum tension. You can't see the individual twitches anymore in this, right? When you get this plateau right here, this is what you call tetanus. And so this is the graph, right? Reading this graph right here, you know, we look at the effect of this stimulation, right? The greater frequency, the stronger force of contraction you can do, and the terms associated with that, right, are wave summation over here, right? That's just the general concept that applies to all of these. But when you have either unfused or incomplete contraction, you have a little bit of contraction in between them. When you have fused, tetanus, right? You have no relaxation in between those stimulation. Because remember, going back to this over here, right? This muscle action potential took about 2 milliseconds, 1 to 2 milliseconds in that latent period. And so during that time, right, you had like, could be 10, 20, 100 times as long as that action potential took, that rather the contraction phase took. So you can fit a lot of action potentials in between that time, so you never get that relaxation. All right, and that is the story of how the nervous system is able to generate more force within a muscle fiber or motor unit. And it does so by increasing the frequency of its impulses such that the sarcomere is never allowed to relax. So at each impulse, the contraction force builds upon the previous contraction force until it reaches full contraction force, which is your fused tetanus. All right, we'll see you next time.